I'm Gordan Lauc. I'm professor at the University of Zagreb and director of the Human Glycome project. I'm also founder and CEO of Genus Glycoscience Research Laboratory. Today, I will tell you something about Glycomics aspects of COVID-19. For billions of years, we, and when I say we, I mean all cellular organisms, are fighting the war with viruses. And glycans are one of our main weapons in this war. Actually, the topology of glycosylation in our endoplasmic reticulum can be explained only if we think about this process being developed at the level of archaebacteria. So when our common ancestor, the archaebacterial ancestor, was trying to evade viruses, it was presenting different glycans on its surface. And this process was happening first on the inside of the membrane, and then when the glycans became too big to transport through the membrane, the assembly was happening on the outside of the membrane. So even billions of years ago, bacteria were trying to become different to evade viruses. Because for viruses to attack a bacteria or any other cell, there has to be a specific recognition. So glycans and some proteins on the surface of a virus would come in direct contact with glycans and proteins on the surface of a cell. And if there is a specific recognition, there can be infection. Viruses in this arms race use gene variants, so different gene sequences, and frequent mutations in these sequences to become different and to recognize ourselves. At the same time, we cannot use frequent mutations because our genetic information is too valuable, so we cannot just randomly mutate our sequence. So we have another weapon in our arsenal, and these are glycans, because glycans are not encoded as by single genes. They're encoded as multiple genes. They're encoded with multiple genes, so they're kind of a complex trait. So chemical structure, which is inherited as a complex trait. And this enables us to create novel structures without actually introducing mutations in our genes. This is something we clearly demonstrated by studying a cohort of collaborative cross mice, which was generated at the University of Western Australia, where interbreeding for only 3 generations generated significant variability in IgG glycosylation so that we can actually group mice based on the IgG glycome, which happened only in 3 generations of interbreeding. And this is IgG, but the same things are happening on a cell membrane. So glycans, which are covering our cells as a thick layer of a glycocalyx. So when we think about the cell membrane, actually the lipid bilayer is this tiny little white lane, while this thick coating is the glycocalyx. Glycans presented at the surface of our cell. And anything approaching the cell, being it another cell, bacteria, virus, or even some signaling molecules, actually have to talk to this glycocalyx layer first. So good analogy to this glycocalyx layer is the thick fortifications which medieval cities used to have to protect from the invaders. So if our glycocalyx is functional, we have these thick walls which are protecting our cells. If it is not functional, these walls do not function. And often these walls are surrounded by a body of water, and this also happens in our cells. Actually, our cells are covered, at least those cells which are exposed to the contact with microbes, like different mucosas or different endothelial cells. They're covered with a thick glycocalyx, and on the top of this glycocalyx, there is a layer of mucin, which consists of water and mucins. And mucins are a specific set of glycoproteins with a very variable glycosylation. So they're presenting very diverse glycan structures, which kind of emulate everything which can be found on a cell. So if there is a virus or bacteria coming on a surface of our mucosa, it's often trapped by those mucins in the liquid layer, and then by ciliary flow is being removed outside of our airways. And this is actually functioning very well, and this is protecting us against all the microbes for hundreds and thousands and millions of years. But this mucosal barrier, this first line of defense against viruses, does not work if you dehydrate the mucus layer. And dehydration of mucus layer is not something which was common during evolution. This actually happened only when we started to live indoor and during winter heat our homes. So in the winter, outside it is cold, and we heat our home so we can have a warm environment. But what happens, this heating is causing the air inside to be extremely dry. So the problem is that absolute amount of water which can be dissolved in the cold air is much, much lower than the amount which can be dissolved by the hot air. So if the cold air at approximately 0 degrees, which even have 100% relative humidity at 0 degrees, enters our house and is being heated to some 25 degrees, its humidity will fall, the relative humidity will fall below 20%. And there has been studies in the US saying that the air in most heated residential and commercial buildings has humidity below 20%. And the relative humidity below 20% means the air is very dry, and the air, when the air is dry, it's actually taking the water away from our mucosal layers. So what happens, mucosa, which is supposed to be well hydrated, dries out, there is no flow of mucus, and there is no way to stop viruses and bacteria from either entering the cells or spreading downwards toward our lungs through the upper airways. Actually, it is believed that dry indoor air is one of the main reasons for seasonality of respiratory viruses, because what we know is that viruses like influenza or coronaviruses, which cause a common cold, are occurring in the northern hemisphere between November and April, and they usually disappear and then come again in the November next year. And actually, it is not only that the people behave in a different way in summer and in the winter, it's also a big element in this story is that air indoors is very dry, which is drying out our mucosa, and we do not have this initial line of defense against viruses. So what are the recommendations is keep the air indoors at least 40 to 60% relative humidity, ventilate it frequently, and there will be less viral infections even in the winter season. But this is only one aspect of the glycomic story of the viruses and the COVID-19. The other one is that glycans are important structural components of both viral proteins and its cellular receptors. For example, if we look at the spike glycoprotein, everything gray here is a polypeptide, color is a glycan, it's actually covered by glycans. And it's very hard to understand function of this protein if we ignore glycans. Unfortunately, many scientists still ignore glycans, simply because it's difficult to analyze them, so they decide to go the easy way and just ignore the glycans. And there was one recent example when even the director of NIH in his blog promoted an image of S-glycoprotein without even a single glycan. And unfortunately, if you try to understand function of this protein, this is not easy, because in biology, this protein functions with all the glycans on its surface. So to understand the biology of glycans, we have to understand the biology of viruses, we have to include glycans in a story. And when you talk about the S-glycoprotein of COVID-19, it has many different glycosylation sites, and these different glycosylation sites can have different glycans attached. And when people study recombinant glycoproteins, they can have very different glycans depending where the protein was produced. For example, if it's produced in the human cells, it will have a very different glycosylation compared to what will happen if the same protein is expressed in the insect cells. But also, when we think about the different people, glycans are responsible for a large part of inter-individual variations. And the best known example are the blood groups. ABO blood groups are glycans in their chemical nature. So glycans is something what makes people different. And if we have viruses produced in these people, and we all produce viruses when we are infected, as well as the receptor for those viruses, they will be differently glycosylated in a different people. And this may cause these glycans to have different functional properties. And one of the key elements where glycosylation is important for function of membrane proteins is the half-life of membrane proteins on the surface. There is a number of studies showing that if proteins have more branched glycans, they will stay on the surface of the membrane for longer than if they have less branched glycans, when they will be recycled faster. And these differences in glycosylation can be functionally very important. For example, increase in glycan branching will be predictive of future development of diabetes and some cardiovascular diseases. And here, glycans are not only biomarkers, they're actually active participants in the development of diabetes, because changes in glycan branching will change the half-life of glucose transporters, of insulin receptors, and other proteins on the cell membrane. Also, changes in IgG glycosylation will change pro and anti-inflammatory properties of IgG, and in this way contribute to the development of diabetes. And if we act early enough, we may be able to prevent these diseases, because normally people go to hospital only when there is something seriously wrong. So when some organs stop functioning, they go to hospital and then they get a diagnosis. But at that time, there are many molecular changes, and this is very hard to reverse. But if we look at the biomarkers, and we can see changes happening much faster, much sooner than the final diagnosis. And if we identify people at risk of developing disease early, maybe a simple lifestyle intervention can help to prevent disease. Something what we have shown recently in a large study is that if you just change your dietary habits, if you lose weight, either by diet or exercise or even bariatric surgery, your glycans can change. And something which we can call bad glycans will decrease, and the better glycans will increase. But this is difficult. People don't like to change their lifestyle. So we would all like to have a magic pill which would help this. And maybe there will be a magic pill, because there are some drugs which are now entering the clinical trials which modify glycosylation. And one of them is this 2DG derivative developed by Moleculin. They call it WP1122, which has been shown in initial in vitro studies, also in animal studies, to be able to prevent viral replication, and at the same time to change branching of glycans on the surface. But this is not the same in every person. So if we want to use this type of inhibitors, we have to think that before using them, we have to do a patient stratification. For example, if some individuals would have a very low glycan branching at the beginning, which can happen maybe in a people on a keto diet, if you try to decrease it even further with this type of inhibitor, they may run into problems. So patient stratification is something which is the holy grail of personalized medicine, including the therapies which would modify glycans. But for decades, focus of personalized medicine is on genetics. People analyze genes. And there was a great technical success of genetics. We can analyze thousands and thousands of people. We can sequence the whole genome for a couple of hundred of euros. But actually, all this technical success did not fulfill expectation of both scientists and general public. There is very little clinical translation of genetic research. And one of the reasons for that is simply the genes do not define everything. And the best example that genes do not define everything are identical twins. And on this photo, we have two pairs of identical twins with every single letter in the genome being identical, but obviously they have a very different life trajectories. And actually only about 20% of longevity and only about 30% of the majority of complex diseases is genetic. Everything else is environmental or combination of genes and environment. And glycans here come in as a complex trait, which is encoded in dozens of genes, but also environmental factors. And this image shows the result of a decade-long GWAS work we did on IgG, which identified nearly 40 genes which work together to regulate IgG glycosylation. So we have a complex genetic network deciding about the structure and the function of single molecule. Because we have to think about the glycans as being integral part of a protein, and actually nearly all proteins have glycans in their structures. So to understand this structure and to understand the function of the structure, we also have to include this colored part, and these are the glycans. Unfortunately, this is very complicated because at least 2,000 different glycan blocks are being added to the polypeptide backbones. And this is increasing the complexity of a glycoprotein tremendously. So we think that the glycoprotein is several orders of magnitude more complex than the protein. And the best explanation for this would be that nucleic acids are built from 4 building blocks, proteins are built from 20 amino acids, and when we add glycans to that, we add over 2,000 different blocks, which can be combined to make novel structure. And this is why glycans are one of the key weapons we have in our fight against viruses. And contrary to polypeptide part of proteins, glycan part is not fixed. So polypeptide is defined by a gene. Once we have our genes, our polypeptide is defined. Glycans are partly defined by genetics, but they're also under effect of environment, including the past diseases, smoking, sleep, exercise, diet, stress, whatever. So all this integrates, all this environmental information integrates through epigenetics to genetic information, and then results in a final glycan structure, which can change with time. And for example, for IgG, we know that when we are young, our IgG glycome is mostly suppressing inflammation, and when we are old, our IgG glycome is mostly promoting inflammation. So it changed during our lifetime. Part of it is heritable, part of it is defined by genes, a significant part is environmentally determined. So the decisions we make in our life are affecting how our glycans will change, and they do not follow some kind of a predetermined trajectory. In a large study we did nearly 10 years ago, we showed that in 4 cohorts of people, over 1,000 people, we see that the glycans follow more or less the same trajectory of changes with aging. So they change, on average, they change in the direction of pro-inflammatory glycans becoming more abundant and anti-inflammatory glycans becoming less abundant. And this is changing structure of the Fc part, and it's directing immunoglobulins to the different receptors. So this is the way how immune system can be modulated. If we put it together in one model, we can actually predict age of a person only using glycans. But there is an error. So if you use glycans to predict the real chronological age, the median error would be something like 9 years. So we can get age plus minus 9 years, which is actually not too accurate. But we are not trying to guess person's chronological age, because this is not important. We know what is the age of a person. What we are actually interested in is what is the difference between the age we measure by glycans and the real chronological age. And when we look for these deltas, for the difference between age and glycan age, we see that this actually associates with the biomarkers of unhealthy life. So people who live unhealthy life will have their glycans changing faster towards the older glycome than people who live healthy life. Also, when we looked into people with diseases, and we studied over 40,000 people with the different diseases, we see that the effect of aging, and these last four columns are the effect of aging, are present in many different diseases. So people with many different diseases would have a glycome which makes them look older. So this is not a specific change in a glycome related to a given disease, but it's a general mechanism, most probably related to low-grade chronic inflammation, which is contributing to development of many of these diseases. And actually, we do see often that these changes in glycans come before the disease actually developed. When we looked around the world and looked for different cohorts from different countries, we realized something which was quite unexpected at that moment. And this is that this difference between chronological and biological age is practically on a line with the expected lifespan of a person in a given geographic region and the Human Development Index, which is measuring development of a country. So the conditions in which people live somehow change their glycans to move faster or slower towards the old age, which then somehow associates with the expected lifespan. So if we think about the lifespan, the primary killer in the world still are cardiovascular diseases. So we checked what happens with IgG glycome and the cardiovascular disease risk, and found in a large cohort of people in UK that IgG glycome strongly associates with the cardiovascular disease risk score, meaning the risk of cardiovascular events in the future. And these changes happen before any other disease symptoms. So practically, by looking at glycans, we get kind of an early warning that there is a disease developing. So we see some kind of biomarkers where people are still without any clinical symptoms, but these symptoms show, these biomarkers show that they're moving in this direction of disease development. So maybe we can do an intervention and prevent disease at the end. And something we just finished recently, again, studying large cohort of twins in 3 time points, we saw that twins who were losing weight were actually aging much slower than twins who had the same age or actually gaining weight. So by weight loss, you can actually decrease aging of your glycome. One of the drastic ways you can also do it is actually taking a bariatric surgery. Bariatric surgery has very drastic effects on a glycome, with some individuals changing this glycan age for up to 30 years. Physical activity can also help, but too much physical activity is actually doing damage and is making your IgG glycome looking older. Based on all this data, David Sinclair and I wondered whether maybe biomarkers of biological age could be a better predictors of severe COVID-19 than only the age, because this is something which is quantifiable, it can be measured either by genetic age or by the glycan age, and by measuring this biological age, maybe we will have a better biomarker for COVID severity in the future, and this is something what we are currently trying to do. And at the end, I have to acknowledge the funding. We have very generous funding from a number of European and national projects, which enabled us to do these all very interesting studies.